So Coral Line's project, how it all came about, I sort of came to Gili 3 years ago. Our house reef is struggling. We've got just around 10% of coral cover on it. It's the primary location for the guests to go snorkel, and obviously we needed to do something about it. Being a large area, it needed specific techniques. You know, just recovering all of that, it's a massive mission. Gili gave full support. The resort really supported me, and I sort of spent several months reviewing literature and trying to find a different way to do it, because I also wanted, as a marine biologist does, to sort of experiment with something new. So I kind of concluded that I need something which is able to recover a large area. It needs to be large scale, and for that you need to optimize your time spent, and you need to use some low-tech methods. So the less technology is involved, the quicker it is, the more reef we can recover. And I also decided to choose the two-step method because we don't have a healthy reef next to ours to take the donor colonies from. So this is our situation. That's Gili Lankanfushi. And that area in a red square, that's our target reef. This is where the guests go for a snorkel. We call it One Palm Island Reef. So there's a little One Palm Island there. And that needs recovery. So for the nursery, because we're going to be using two-step method, we decided to use the sort of lagoon area there in the blue. There's a little platform there which helps us to actually access it with the guests. So a little floating platform really goes well with the project. So the idea was to recover that reef. Also being a resort, we needed to make sure that it's a guest experience. But at the same time, apart from just sort of showing the guests the planting process and all that, we also wanted to engage them in the science. And by sort of integrating the data collection procedure into the guest experience, we made sure that data does get collected and we can do research on it later. So also our focus was research, not profit. So when the guests do donate Coral Line with us, 75% of it goes into our Gili Seas Fund, which is Social, Environmental Awareness and Sustainability Fund. And that's used for coral lines and also for social and environmental projects. The whole research purpose was to find an effective way to recover a reef fast. As you'll find out later, the rope method that we use has been practiced around the world. However, nobody knows how to transfer the ropes back to the reef. And that's a big challenge and also a big opportunity for us in the project. And yeah, Debs came and joined me a year ago, and we developed on the concept further, and we decided that
we wanted to share our findings. So we wanted to make it nice and open access. You can see our website in a little bit, and everything is on there. All of the data that we're collecting, we really trying hard to put it on our website, which is open to everybody.
Yeah, so idea is not to say, you know, we're doing this and look, it's great. It's more about, we're doing this, if you like it, join and do the same thing. So we're happy to share everything. Yeah, that's the guest experience bit with data collection. I forgot to change the slides, and this is our website. You probably have visited that. So we've got a video here to illustrate where we are now, how we got there, just to keep you guys awake as well. And just to mention, the rope nursery method is not our invention. We literally did what Levy et al. In 2010 did. So it's the same type of nursery, exactly same parameters, everything's the same. I don't know, there's a bit of music there as well. So we started off, that was nearly 2 years ago. We welded some angle bar together to produce the platform of the nursery. Dunked it in the lagoon, and then it was a challenge of hammering it in the sand. That took a lot of effort, some improvised methods, and we could get about 2 frames done in a dive because we would run out of air. It was difficult. Also aligning it was difficult because the visibility is not always great. Eventually, somehow, we got it right, and it did align. So as we mentioned before, the basic principle of this nursery, elevating it from the sand, means that you get less sediment. And also you don't get corallivorous animals eating the coral. They can't access it, so it ensures that small corals are not damaged. Then we started collecting corals of opportunity. We've got a lot of loose rubble on our house reef, and a lot of the table corals get flipped upside down, so they would die anyway. We collect that and we keep it in a nursery area. This is our platform that we use for the guest experiences, and that's how it works.
Here we are with the guests. So what we do is we select a coral, we fragment it using various tools, hammer and chisel, but more recently we have started to use bone cutting pliers, which are being used here. Really, really easy way to fragment the coral, if any of you haven't tried this. So once we've done that, we start to put the pieces into the rope. So that's the first one. So we put 50 fragments of coral into a 5-meter length of rope, and they're spaced evenly 10 centimeters apart to allow for some growth. But because they're all from the same genet, even if they grow big and they actually touch, they'll just fuse together. So the guest really enjoys it, getting up close and personal with their coral. And there we go. So we measure them as well. Every single fragment is measured, and the guest is involved with that collection of data as well. So that's line number 1, the first line we have planted. Ready to go.
And we snorkel with the guests as well. We take them above the nursery. They hold one end. We free dive down, as you'll see, and attach it to the nursery.
So we use cable ties at each end of the rope to attach it into the metal frames, which have got holes drilled in the top.
And then the first year, obviously, was literally quite ropey sometimes. We had various little challenges. But one of the things that helped us a lot is the database that we've developed beforehand. And this is what processes all the data for us. And then we just have the website to update, and the guests see this.
So what we do is sort of once a week or so, we go for a maintenance dive. And this platform that we have in our lagoon is right above the nursery, so it's really useful for us. We clean the algae off the corals to avoid the algae sort of smothering over.
And we had various interesting things with this algae and the cleaning itself, as you'll find out later. We had a bit of an outbreak of disease as well.
You see what we did, we actually cut off the diseased parts of the coral, and it seemed to work quite well.
And the measurements. Every 3 months, every single fragment again re-measured. It basically means that every fragment is a separate sample. And one rope itself can work as an experiment because there's 50 samples.
So we waited for them to grow.
And we waited and we waited. And they grew. So this is 3 months actually. This is various different ages and different types of coral.
So we currently have 20 different species in our nursery.
And yeah, that's the nursery today with more than 2,200 corals planted. And how many we've got left? About 2,000 at the moment.
Yeah.
And that's our target reef, which is awaiting transplantation. So that will begin this year. So it's a big, big challenge. Cheers.
Thank you.
Thank you. So let's look at where we are now with the transplantation.
So obviously, one thing that we've obviously left out of the video because we haven't done it yet is the transplantation part. So it's quite exciting. Lots of experimental ideas going on here. We have a 1-year grow out in our nursery. So the lines that have been in for a year, it's time for those to be transplanted. And we've thought of a few different ideas. You can see there's a drill up there and sort of U-shaped nails. What we wanted to do was actually nail the ropes down onto the reef. Originally, we had to go and we tried it with a hammer, and it was really quite hard. So we decided instead to grab a drill. This is just a normal pneumatic air drill, and you can use it with the right equipment, attach it to a scuba tank, and drill into the substrate underwater whilst you're diving or snorkeling. What we wanted to do was drill little holes, get our ropes on the reef, and nail them down, and really cement it in with some marine epoxy or something like that, making sure it's nice and sort of solid. But it's actually a step that our guests are really quite excited about as soon as they find out that it is an experiment and we don't really know what we're going to do with it. We've had guests waking up in the middle of the night and writing things down to tell us about it. They've spent more than 2 hours in the dive center giving us all their exciting ideas. Quite entrepreneurial, engineering type guests, some really interesting stuff, and some of it we're actually going to use. So we found that as a guest experience, it's also really great, and they really enjoy the science side, which actually really surprised us.
Yeah, we were really worried about it. We have disclaimers saying that this is a scientific project, and we were worried that the guests will get upset if something dies there, or if it doesn't work. But when we present it to them as an experiment, they actually get engaged themselves. They say, oh, well, I want to help. And we had guests who said, I'll plant a line each year, provided that you use my method. And it's quite interesting.
So one of the main papers that stood out during Vaidotas' literature review at the beginning was this one by Lindahl, where he actually placed ropes on a reef, not using a two-step method. He just he put the ropes in the coral, the coral in the rope, and just put them straight on his target reef in an effort to stabilize the current substrate and the corals themselves. And for him, it really worked really well. So we thought, we have the same pretty much sort of system, only we keep them in a nursery for a year. So hopefully for us, it should be just as successful. So we're really aiming to crisscross our ropes, and because on our target reef we have a lot of unstable substrates, it's hopefully going to nail that down. And we actually have quite a lot of recruitment on our reef. We've got a lot of really small corals that just get flipped. So we're hoping that by stabilizing that with coral, it's really going to help recover much faster than it would do naturally.
So this is what our guests get to see, the pictures, the picture bit. Line number 2 here with Acropora hyacinthus, very, very successful. I think only 2 or so fragments have died on that. This is the start with very small fragments, some under 3 centimeters, and that's the same line 6 months later. You can see there's a little bit of growth, and then 1 year later, there's quite a lot of growth, which surprised us actually. And also the guests don't only get to see that, they also get the basic data for them. So that's the growth, well, the sizes of the coral, and that's the survival. So how much what percentage of the coral fragments is still alive on that rope. And by putting this on our website, by having it as a communication with the guests, it puts us in a position where we have to do it. So the data collection has to happen, and we have to do research, which is good.
So moving on to what we actually collect. So we collect size data, so we do measure our fragments over time, which allows to give us sort of a growth rate we can calculate from that. And also the survival of the corals. So obviously as we're going and we're measuring, we're noting down which ones are alive and which ones are dead. So from that we get survival rate as well. Quite simple methods, and I'm sure it's what most scientists will do in their nursery. They're quite easy things to measure. But for the future, obviously, the transplantation success onto the reef with different methods is something that we're focused on. But also things like necessary nursery maintenance. So we were actually spending quite a long time at the beginning cleaning up lines and things like that. And we wanted to know exactly how much time do we actually have to spend doing that. Does it really matter? So quite a lot of ideas that can actually come out of this project, which is quite nice.
And very, very important bit that needs to be mentioned, quite boring to be honest, because you're going to be looking at this. But it took months to actually prepare the full database for the coral lines. We used Excel, and it took that time. However, now we're ready. We take about 10 minutes underwater to measure one rope with 50 fragments, take 50 samples. It takes another 5 minutes to put it in this Excel sheet. But the Excel sheet is designed in a way that it calculates everything by itself, the mortality, the growth itself, and then that sheet is also linked to a master database which tracks everything, including the actual dates of monitoring. So since we had this prepared, it saves us a lot of time now. We don't have to worry. We just put the line in, enter it into the database, and worry about it when it comes to monitoring. So that helps us a lot, and obviously, we do advise if anyone's planning to start a project, get this ready before you begin. Plan it out, do the design first.
So a bit of a visual, our size data. So we do measure our corals every 3 months. This one is line number 6. This is a Acropora aspera. It's one of our fastest growers. You can see at the beginning here, number 6, we've got an average size of fragments, 53 millimeters. So it's quite small, about 5 centimeters.
Yeah.
And after 9 months, the growth rate's actually quite fast. And we've got more than 200 millimeters on average on that line now. Which is quite surprising. We were actually surprised at how fast this species was growing. But we do have a whole range of other species in the nursery as well. So on the next slide, we've got a nice graph for you. We actually have 20 different species. This is just the species that have been monitored at least once to give a nice sort of sample. But you can see there's 2 clear front runners. These are the 2 staghorn species that we've got in the nursery. So we've got Acropora muricata and Acropora aspera. So you're nodding your head, you also agree we've seen the same thing. What we've actually done is we've planted another one, Acropora pulchra, which is another sort of branching one as well. And we're interested to see if that's very similar. But as we expected, there's quite a lot of slow growers as well. So you can see a lot of the postuloporids and porites as well we've got on the lines. They do grow relatively slow, about 5 millimeters, and that's per 3 months there. So that's sort of almost 5 centimeters per 3 months for our aspera, so pretty quick.
Also, this graph might actually be substituted for another one later on in the project, because what we found is that the coral doesn't grow linearly. It kind of does it exponentially. So as the colony gets bigger, it grows faster. That's so far from the sort of data that we've got, but we're just waiting to accumulate enough 1-year-old lines to actually decide whether it's this way or not, and run some stats on it. But this is just an indication of what happens on average per quarter. And yeah, some extremes here. Obviously, branching corals versus massives, something that we expected to find anyway, is massives grow slower, with slowest ones being porites, but they do extremely well. And then there's the aspera that grows really quick. Now, in terms of actual survival, was it your slide or my slide? I can't remember now. Right. So we're looking at which species. Initial idea was to see which species do well and which don't on this particular nursery type. To our great surprise, we see very, very little initial mortality associated with stress or with this particular method. Most of the corals did exceptionally well, as you'll see in the next slide. The only exception was this Acropora selago, which was very fiddly, and we damaged it a lot while planting with the guests. So that was the only real exception. That's in terms of initial survival. And what we found later, as we ran the project for a year, is that some corals are more prone to disease. So this graph explains it all.
So we've got our survival rate here on the top left. This is we've got 82% survival, and that accounts for all of the corals that have been monitored for at least 3 months. So that's that if we were to count all of our corals, we would have 85. So it does change, it is relative. But here we've sort of grouped our species again. And you can see sort of the percentages along the bottom of survival that we've got per species. Some of them to note, the top one, Acropora aspera, again, a fast grower, but also it's doing particularly well. More than 200 fragments of aspera in the nursery, and there's only 13 that have actually died, so we thought that was quite impressive. And there's a couple of others as well. We've got what have we got? We've got Hyacinthus, which is quite an interesting one. There's about 300 fragments of Hyacinthus in our nursery. Only 27 of those have disappeared and died. So that was quite an interesting one as well. And one of my favorite ones, which I'm finding very interesting at the moment, is Acropora humilis. And it seems an extremely resilient species. We've planted just over 160 fragments. And you can see there's 7 up here that have died. But that wasn't actually natural death in the nursery. That was a mechanical malfunction where the cable tie actually broke, and the line fell into the sand, and 7 of the corals then got smothered and died. So I fully believe if that was happy and it stayed in the nursery as it should have done, we would have had 100% survival on that one. And we found it doesn't get diseased, it grows very nicely, it's a nice one, it's very, very interesting to see.
So for the most of this, it's actually pretty good. I mean, we've got 90s for most species, 90% survival or so. So the ones that didn't do so well, we found particularly Acropora digitifera, although we only have 2 lines with about 100 fragments. That did worse, actually. It was very susceptible to the disease and died sort of in the period from 3 months onwards. As it got older, more and more of it died. Also, we had some issues with disease with Acropora muricata and nasuta, and then the Selago is the exceptional one, which suffered from initial mortality. So for the most of this, very, very good. And we were extremely pleased with the results.
So some of the challenges that we faced, we thought it was quite important to share with you some of the problems that we had. I don't know if any of you guys have faced the same ones. If you've got different solutions than we've come up with, let's see. One of the main ones was disease, and it's something that I've heard from various nurseries, people working in nurseries around the world telling me that disease is probably one of the biggest issues. Some kind of white syndrome here, which is probably bacterial. And it sort of appeared out of nowhere one day on quite a lot of our fragments, and usually on the same lines. And what we thought it could have been was actually our intensive cleaning method. So we thought we were probably causing it and actually spreading it as well. We were told to use a toothbrush to clean our nursery to make sure that the algae is clear from the coral. So it's something that we were doing, but using the same toothbrush for a whole line and for several other lines as well. We've probably accidentally abraded the coral tissue, which sort of causes an opening really for pathogens to enter. And then we've obviously spread whatever it is, and we've ended up with quite a lot of disease in the nursery. So it was quite a big problem. Another thing that sort of backed up our theory was that, as you can see on this particular coral, the dead tissue, the disease actually progressed from the rope onto the coral, which is probably from us cleaning again. That's what we thought it was. And we sort of did a bit of a test, and we just stopped cleaning it. We just left it alone. And you'll see in the next slide sort of some of the solutions that we did. So we didn't clean it as much. And we actually started to cut off, particularly in the branching corals, the disease bits. And this idea actually came from a guest who had corals in an aquarium at home. And when we looked at sort of aquarist websites and forums, it's something that quite a lot of people will do. Using bone-cutting pliers, we bought these from a vet, and actually just cutting off the disease so that it doesn't spread further along the coral. And it actually worked. And we've got some good pictures for you as well. So this one is a Acropora muricata. And you can see the white, that's dead skeleton. The brown, that's live tissue. And you can see it is starting to progress. And what we did is we cut that branch right off.
Bit of a messy operation.
We actually made quite a mess of it. You can see it's quite bad. And we were pretty sure that's probably going to die. We've pretty much nailed that. But to our surprise, it actually started to grow back. You can see a couple of corallites there. And over time, we started to see more and more growth. So we were actually really surprised. Even little axial corallites starting to come back as well. So it does work, not in every single case, but it's something that you can try if you're having the same issues.
It worked for most branching ones, but for digitifera, it didn't. It's impossible. It's really tough skeleton, and we couldn't do the same with it. So yeah, algae, nevertheless, we stopped cleaning, but it can still be a problem. It sort of algae starts growing around the fragment, and eventually just covers it completely if left out of control. So there you can see there's brown stuff growing around it. But what we do now is we just shake it off, as you saw it in the website. It took us a while to stop the toothbrush method. I think we began it actually because we were too scared that we're going to lose all the fragments. So we're like, okay, let's clean as intensively as we can. Then we sort of fought with ourselves about stopping. Eventually we said, no, okay, no more. And yeah, it all recovered. So now we just shake it off and waft water. And also the fact that the line is a flexible structure, you can actually use it to shake off the algae, which helps us a lot as well. So we just sort of move it around, and that detaches the algae.
So some of the other issues we had, so we've all had equipment malfunctions. We've had some problems with measuring, in particular was one. Our calipers that we measure with, they're actually not big enough. We really didn't expect the growth rates that we saw, especially with Aspera. And the only calipers that we could find to buy online were 6-inch calipers. And our corals are actually too big now. So what you might see on our website, if you do have a look, is sort of just a bigger than 200 millimeters, because that's as much as we can get to. So that's a problem for us, trying to find calipers that actually are big enough. And another thing that we were thinking of is that the calipers themselves, is it actually the most accurate way to measure size? This coral here, you can see there's a few different ways you could measure that. So what we've been doing is taking what we believe is the colony diameter, which is the widest point of the coral. But sort of something like this, should we be counting the number of axial corallites? Should we be, taking maybe a surface area using some software like ImageJ or something like that? It's not the most accurate way to measure. It gives us a measure, but there's definitely somehow a better way to do it. So it's still something that we've not managed to do yet, but
it's quite interesting. One guest got so excited about it. He said, "I'm setting up a cylindrical tank with a camera system basically on a rail, and the camera moves around, takes pictures 360 degrees of." He's an IT guy, and basically he's going to model three-dimensional corals and get the growth rate that way. So he should be actually coming back in March, so it should be interesting to see.
Yeah, but basically what we wanted to do was just make it as easy as possible, because we've not got all the time in the world to go measuring in all these complicated ways, as I'm sure most of you also have. So we wanted to find an easy way and really streamline the project to make sure that it is quick and that we're getting the data, but we're not spending ages doing it. So a few ideas. The drill as well that we bought, I don't know if any of you have a drill or you've used one underwater. At my old island we had one, and I've heard various other people using it as well, but we weren't really sure how to go about acquiring it, what equipment we needed, was it safe, and we actually utilized Coral List for this. We sent out a big message to everyone on Coral List, and if any of them watch, thank you for helping us. But yeah, they sent us a whole lot of really interesting data, different scientists that have used it for different reasons and different methods. And we sort of pooled what we got, and we came up with what we thought would be the best method for us. So we bought a drill, and then we came into Male, and a really nice guy from a shop rigged up a special regulator for us to make sure that the air pressure was right and we weren't going to sort of explode our drill. So that was really helpful. And then finally, things like cable ties breaking, really small issues that we've had in the nursery. We cable tie our ropes to the metal frames, and after so many months, 9 months, 1 year, they get sort of encrusted upon, and they break. And then that means our rope is in the floor, and the corals are dying. We've had about 3 ropes, I think, that's happened to. So it's not a huge problem, but it's there. So there's possibly a better way to attach them. We've started doubling up the cable tie, so it's got a little bit more sort of pressure there, but again, sort of questions without answers.
So for drill, watch our website. As we start using it, the information should appear. So yeah, last bit about the project is just highlighting the fact that it is a time when it's really easy to share information. Our whole project is based on Google Blogger platform, which is free to use, and you can actually make a website out of it as well. So that's what it works with. It's very easy to use as well for those of you who have never sort of done any internet web page design. It's got templates. You just pick a template, add some images, and you've got a website. So it's easy to share the information. It's never been easy actually, and it's for a good cause as well. Because I'm sure every single marine biologist knows something that we don't know, and so on and so forth. And if all that information is, shared. Then that can contribute to the great benefit of all. So,
yeah, so we basically, when we were doing our project, we were obviously looking for answers, and we were sort of thinking about how to do certain things, and obviously we faced a lot of problems. And we just sort of thought, what if everybody shared, someone's done this before, if that was out there and we could read that, our project would be more successful, less corals would die possibly. And things like that. So it really is useful for all of us to share and actually benefit from each other's experiences as well. So what we really wanted to have a huge focus on today was really trying to get it out there that we should all sort of make some kind of platform so that we can actually share what's going on and we can learn from each other as well. It's quite an important thing, I think.
Yep. And as for us, we're happy to share everything pretty much. You know, get in touch with us if you need templates or whatever you. So just get in touch with us via email, and we'll be happy to provide you with some things that we've developed ourselves and some ideas. Our website, we decided to save paper, so there's no handouts today. lines.lankanfushi.com is our website, and I've given you Deb's email, which you can contact if you want extra information. I think there's also going to be a...
We've got a sign-up sheet as well, so if any of you guys leave your email for us, we will send you our presentation as well from our email, so you've got that contact there as well.
Yep, so our reference is what we use. There's the big reef rehabilitation manual, the big book about restoring reefs, also the Reefs at Risk Revisited, the big review about the status of the coral reefs of the world, and those are the two specific methodologies that are employed in our project. And that's really it from our side, guys. Thank you.